Our world is solar powered. It all begins with the sun. The sun's rays slowly make their way towards Earth, each with their own set of wavelengths and frequencies. The higher the frequency, the shorter the wavelength, resulting in more energy, and vice versa. The visible light spectrum has wavelengths from 380 nanometers to 700 nanometers, which is light that is visible by the naked eye. First, we will take a look at the reactants of photosynthesis and how the plant obtains them. We start with gas exchange. Carbon dioxide enters, and O2 exits via the stomata on the lower epidermis of the leaf. Guard cells, which aren't pictured, can open or close to control the evaporation and rate of exchange. Next, we will look at the impact of visible light on the leaf. Each of the different wavelengths on the visible light spectrum has different effects on the rate of photosynthesis, and each type of chlorophyll is specialized in taking in one type of light. This can be seen through the action and absorption spectrums of light. Green is not used in photosynthesis, so the plant reflects the light off, giving it the green color. Last but not least, a large central vein delivers water to the leaf through the xylem while taking glucose and the products of photosynthesis through the phloem for storage, usage, or plant growth. Now these are the light-dependent reactions in photosynthesis. The water gets split by photosystem 2 into 2 hydrogen ions, 2 electrons, and 1/2 oxygen. The electrons then get energized by photosystem 2 and go through the ETC and fuels active transport of H+ ions. After getting unenergized, they go to photosystem 1 to get energized once more, and both the electrons and the hydrogen ions attach to NADP+ to make NADPH, which is used in the Calvin cycle for energy. The concentration gradient of H+ ions leads to the facilitated transport through ATP synthase, which generates ATP for the Calvin cycle to use. Moving on to the Calvin cycle. We zoom into the chloroplast to find the NADPH produced in light-dependent reactions and follow it through the thylakoid membrane. The Calvin cycle starts with 3 carbon dioxide molecules. They are then combined with 3 RUBP molecules by the enzyme Rubisco. The new 6-carbon molecule is unstable, so it quickly gets hydrolyzed into 6 3-PGA molecules, also known as 3-phosphoglycerate. The 3 PGAs are then phosphorylated. Then electrons from NADPH are added, breaking off the phosphorus groups, turning the 6 3-PGAs into 6 G3Ps. 1 molecule of G3P leaves the cycle as half a molecule of glucose, and the other 5 are rearranged by 3 molecules of ATP back into 3 RUBP molecules, ready to restart the cycle. The cycle must then repeat for 1 more time, and then after some more enzymatic reactions, we get 1 molecule of glucose. Now we're moving on to cellular respiration and glycolysis. First, the glucose is phosphorylated by 2 ATPs, then broken down into 2 G3Ps, and 2 phosphate groups are then joined on by 2 NAD+ groups, reducing it to 2 NADH total for both G3P molecules. The 2 phosphate groups are then taken by 2 ADPs, turning them into ATP. The molecules are rearranged and a water is released from both molecules. Then 2 more ADPs come and take the remaining phosphate groups, creating a net total of 2 ATP, 2 NADH, 2 waters, not pictured, and 2 pyruvate molecules. Krebs cycle takes place in the mitochondrial matrix. It is split into 2 parts, pyruvate oxidation and the actual Krebs cycle. In pyruvate oxidation, 1 molecule of pyruvate is oxidized and loses a CO2. Acetyl-CoA is formed by the joining of coenzyme A. For each molecule of glucose, this cycle runs twice. The Krebs cycle is what it sounds like, a cycle. For simplicity purposes, the only thing depicted now will be the carbons. First, a 4-carbon chain called oxaloacetate is added to the acetyl CoA, which is hydrolyzed using a water molecule produced from glycolysis, breaking the coenzyme A and sulfur bond away, making a 6-carbon molecule called citrate. Citrate is then oxidized and loses another CO2 molecule, forming alpha ketoglutarate. This new 5-carbon molecule is then oxidized and loses another CO2 molecule, as well as producing 1 ATP, which forms succinate. Succinate is then oxidized into fumarate, which generates 1 molecule of FADH2, which is then oxidized as one last NADH plus is reduced to NADH. One turn of the Krebs cycle is completed with the regeneration of oxaloacetate. There are 2 turns for every molecule of glucose. Oxidative phosphorylation takes place in the inner mitochondrial membrane. NADH undergoes oxidation and loses 2 electrons and 1 hydrogen ion, which allows for the active transport of the hydrogen ion across complex 1. This is the same for FADH2, which also undergoes oxidation and loses 2 electrons and 2 hydrogen ions, which allow for the active transport across complexes 3 and 4. This active transport is possible due to the ETC, and the electronegative oxygen at the end attracting the electrons down the transport chain. 2 electrons then attach to the final electron acceptor, oxygen, along with 2 hydrogen ions to create water. In this case, there are 2 water molecules being made. In the ATP synthase, because of the concentration gradient created by the ETC and the active transport of H+ ions, they allow for passive, facilitated transport across the ATP synthase protein, which allows it to turn and produce ATP by attaching a phosphate onto ADP. In total, oxidative phosphorylation makes around 30 ATP.